All right, hi everybody. My name is Sam. I'm a fourth-year PhD student in ecology evolutionary biology. It is my pleasure to introduce this year's graduate student invited speaker, Dr. Colleen Iversen. Colleen is actually from Michigan originally, where she attended Hope College for her undergraduate degree, and then went on to obtain her master's at Notre Dame, followed by her PhD at the University of Tennessee, working with our very Henry Klassen and Richard Norby. While she was there, she was also working at the Oak Ridge National Laboratory, where she's continued her work, first as a postdoc and then as a staff scientist, starting in 2010. And from there, she has worked her way up to now be a distinguished staff scientist as the group leader of the Plant-Soil Interactions Group and the director of the NG Arctic Project. She's also the lead PI of the Fine Root Ecology Database, and the former lead PI, one of the lead PIs on the below ground task components of the Spruce Project. She's also a member of the inaugural class of the New Voices cohort of the National Academy of Science and an early career fellow at the Ecological Society of America. I'm very excited to hear her talk, so please join me in welcoming her.
Thanks, Sam. Thanks, everyone. Can you hear me okay? Is the mic placement good? All right, awesome. Thanks for the introduction, and thank you to the graduate students for the invitation. I was saying I hardly ever accept invited talks anymore unless they are offered by the graduate students, because I think that's my favorite interaction, and it's also super important. And you have a great department here. I've never been to a graduate student speaker lunch where 30 folks showed up, and so I really have appreciated all of the conversations. So thanks for that. And no pressure to give a talk in front of a previous PhD co-advisor, but it's been lovely to see folks from there. So today I'm going to give you a little bit of a tour, and the sort of theme of the tour is thinking about the hidden world beneath our feet, and how it can shape how ecosystems respond to climate change. I used to be big on Twitter, and then and so now I'm trying out Threads and also Blue Sky, but you could check me out in both places and let me know if there's a better place, because important science communications happen, I think, on social media. So it's just my name here on the title side, but of course this work is done in collaboration with many, many amazing people, and I have photos throughout to sort of point those folks out to you. So, let's see. Uh-oh, George. There we go.
It's a little slow.
Me too. So I am an ecosystem ecologist. That means I use a variety of tools and techniques to understand how ecosystems are shaped by climatic change. I'm interested in understanding the cycling of carbon and nutrients between soils and plants. And as you'll see from today, I do that in many places in the world, including temperate forest, including squishy peatlands, and including increasingly in the Arctic tundra. And you might notice throughout the talk that I'm a strong advocate for the below ground world. I'm particularly interested in the tangling of plant roots with the surrounding soil. And that's in part because it's a part of the world that folks don't think about very often, and a part of the world they don't get to see. And I like to be able to see a part of the world that folks may not have in the back of their mind. But I'm also interested in what's happening below ground, because soils hold twice as much carbon as the atmosphere. And if you want to understand the importance of soils, you can listen to the TED Talk by my friend and colleague Asma Asfaw-Berhe, who was until recently the head of the Department of Energy's Office of Science, who has a TED Talk with the title, "Stop Treating Soil Like Dirt." But of course there are ways to peer into this below ground world, and some of the work I do is using specialized cameras called mini rhizotron cameras to take photos of roots and their fungal partners. So you can see these roots that are narrower in diameter than this cord that's connecting my microphone to the battery. And you can see that it's not just roots who are making a living below ground, these fine roots taking up water and nutrients to keep alive these towering trees above ground. But they also make partners with fungi that are showing up here as these little white fuzzy bits on the roots. So on the left there is sort of a spruce root, and on the right here is a larch. And I also think it's just beautiful to get to see what other folks don't get to see very often. So the overarching question of my research, down in front, is how the interactions between plants and soils might change in response to changing environmental conditions. So today I wanted to take you on a little trip from squishy bogs to frozen tundra and a couple of places in between. And I wanted to sort of use this map of North America as a guidepost. This is my favorite map of North America because to me it looks like soils. I think it was meant to be concrete, but we're going to pretend that it's soils. And so we're going to start here in Tennessee, where I did my PhD work in a temperate forest. I'm exposed to elevated carbon dioxide concentrations. And then we'll move further north and further into the present day to Minnesota, where we're looking at the impacts of warming and elevated CO2 on a peatland in northern Minnesota. And then even further north to Alaska, where we're working in a couple of different places, Utqiagvik, which used to be Barrow until 2016, and also Nome, which is the endpoint of the Iditarod. So first, starting in the past, in Tennessee. So I wanted to mention, since a lot of the questions today have been from graduate students about what it's like to work in a national laboratory, sort of the history of this large-scale, long-term thinking that's happened at Oak Ridge National Lab. It's nice to be at a place that sort of has this history of systems thinking, since I'm an ecosystem ecologist. And so I started in the environmental sciences division with radio ecology, which I think you know why, thinking about radio ecology back in the 50s there. But moving into thinking about systems ecology, and then part of the IBP, and then thinking about climate change through acid deposition, and then thinking about models, which will be a little bit of a theme of this talk too, this individual-based models, landscape ecology, and then in recent years, thinking about how we can manipulate ecosystems to give us a window into what the climate of the future might be and how that's impacting the carbon cycle, which is something that the DOE, Department of Energy, who funds all of my work, cares about. So my dissertation work was done under Rich Norby, who some of you know, on what's called a free air CO2 enrichment experiment, which ran for 12 years and was in our backyard in eastern Tennessee. And it was thinking about how the eastern deciduous forest might respond to rising carbon dioxide concentrations in the atmosphere. And so for folks who are interested in that, instead of studying a sapling in a pot or seedling in a tumor, we're able to expose entire intact forest plots to elevated carbon dioxide concentrations. So these two rings here are 25 meters in diameter, and they are exposing these sweet gum trees to carbon dioxide concentrations that we expect at 2050, in the year 2050, so about 565 parts per million. We had two rings that were just blowing ambient air, which over the course of the experiment changed in concentration. And then one ring here that was a control plot that wasn't receiving any blowers. And so you can see the scale of an experiment like this. This is sort of a real road. And for someone like me, who's interested in studying the interface between plants and the soil, it was very cool to get to study what was happening in an intact forest system in response to rising CO2 concentrations. And so this was a newspaper article from 2002 where someone came and they sort of drew all of the things that were being measured within the forest stand. And so I like to include it because I think it's a cool picture. And so we were measuring the response of what was happening in the system at all different scales and all different sort of types of responses. So soil respiration, what was happening below ground with these roots, this camera that I was telling you about, which is a clear tube that you insert a camera into and take pictures of root birth, growth, death over time, measuring growth of the wood, measuring what was happening above ground with light and the wood and the leaves, measuring weather, and then you can see sort of the CO2 release from these vent pipes in the forest stand. And what we found was, contrary to what we expected, I say we, I was 8 years old when this started, which I like to remind much of. We found, contrary to what we expected, we thought the trees would grow bigger and taller, more wood, fatter. We found increased root growth in response to elevated CO2. So plants take up carbon dioxide via photosynthesis and use it to make more plant parts. That can be roots or wood or leaves. They made more roots, and that's because plants are not only made of carbon, they need nutrients to grow more biomass, right? And so these roots were increasingly grown and deeper into the soil to acquire nitrogen, which impacted the amount of carbon stored in the soil, and ultimately sort of limited the response of this forest to rising CO2. You can see that here. So in the first year, we saw the expected response. We saw increased production, so anything above this 0 line is greater production under elevated CO2, and net primary production is the total amount of organic matter produced over the course of 1 year, and I've subtracted ambient from elevated. The first year we saw most of the extra carbon fixed via photosynthesis allocated to wood, right? But because the Department of Energy is willing to fund long-term experiments like this, we were able to see the change in the response over time. So increasingly, that extra carbon fix was allocated to the production of fine roots to take up water and nutrients to grow bigger trees. But you're thinking of the wrong kind of roots. So not big and woody like the spicus tree in Hawaii or coarse roots that you might trip over when you're walking around in the forest, but tiny and short-lived, often only for the span of a year. So these roots that do the hard work of water and nutrient acquisition for the plant are tiny but mighty. And those are my fingers, so obviously not to scale. So the interesting thing about fine roots is that there's a lot of heterogeneity. Things change across root orders, which are ordered like streams. So you have your first, second, third order roots. You change anatomy, morphology, chemistry, architecture, mycorrhizal colonization, which changes physiology across those orders. So it's not just a homogeneous pool of roots. There's a lot going on, which led Kurt Pregetzer to write in 2002 that the fine roots of perennial plants are a royal pain to study. He wrote that in a New Phytologist article, and it's true, but it is a labor of love. So moving more into the future and further north to Minnesota, so the Department of Energy asked us to move out of our backyard and not study the temperate deciduous forest anymore, but to move to ecosystems that were understudied, but important parts of the global carbon cycle. And so for us, that was peatlands, which only cover 3% of the global land surface, but store 1/3 of global soil carbon in deep deposits of peat that have accumulated for millennia since the last glaciation. They also asked us to do experiments that were no longer single factor. And so the spruce experiment is looking at elevated carbon dioxide, but we know and care about rising atmospheric carbon dioxide concentrations because it's an important greenhouse gas, which will warm the atmosphere. And so we wanted to look at the impacts of warming on a system. And so Paul Hansen is the PI of the spruce experiment, and we built these large chambers in northern Minnesota, and I'm going to show you more pictures just for scale. But instead of it being free air, as we were able to do with the FACE experiment, we haven't yet figured out a way to do free air warming, and so we had to use open-top chambers, albeit giant ones, to warm up these systems. And so you can see sort of laid out in this bog in northern Minnesota, we have 10 plots. 5 of them receive a range of warming from 0 degrees Celsius to plus 9 degrees Celsius all day, all night, all year round. That's plus 16 degrees Fahrenheit all day, all night, all year round. And then we have another 5 chambers that are also receiving that warming regression design plus 900 ppm CO2. And you can see one of the cool things I was telling folks about working in a national laboratory is that you can really leverage some of the engineering that's possible there. So the FACE apparatus, these chambers that were designed to warm below ground with heating rods that warm 0 to 3 meters around the outside of the plot, and then just 2 to 3 meters inside of the plot so you don't have hot spots in the rooting zone, which folks like me care about, and then warm air cycling above ground. And this is what the bog looks like from an aerial view, and there's a sign here above this plot says welcome to a warmer future, and this is a real door, so you can sort of see the size of these plots. And again, this was necessary because we want to understand the response of an intact, in this case, bog ecosystem to warming and elevated CO2, and so we built this apparatus around the natural bog ecosystem. So just thinking about sort of my interest, uncovering the secret lives of these fine roots in saturated organic peat presented challenges and opportunities for our team. And so I just wanted to show you a little bit of what the soil looks like in a bog. So a bog is dead, decaying moss from 0 to 3 meters or 4 meters or 5 meters. And it is basically roots of this like ericaceous shrub here given this dead and decaying moss a big hug below ground. And that's what the soil looks like. So instead of sort of sieving roots from the soil, you are untangling them from the soil. And the first thing we wanted to do is to understand where the roots were at in the soil profile before we started the experiment. And so the way that bogs are, they have this sort of undulating topography. They have hummocks that are raised and hollows that are a little bit depressed. And they have a high water table, the light blue there. And that's because they're saturated, and that's what allows them to accumulate peat. They're wet, they're cold, and they're acidic, and that's why this carbon below ground isn't decomposing. And I feel like this slide is a really nice example of that. And so we took cores to 2.5 meters and untangled roots from throughout that depth profile. And we found roots throughout the depth profile, and I was like, what is going on? These roots should not be growing below the water table level. We had them carbon dated, and I was holding intact shrub roots that had a carbon age of 4,000 years or 5,000 years or 6,000 years. Have you heard of the bog bodies, right? I was holding the bog body of these roots that weren't decomposing, and the living roots that are taking up current atmospheric carbon are constrained to above that water table level. So what we have found after we started the warming in 2015 is that warming had strongly increased the nutrient availability in the peat, which has also been found in uplands, and also the growth of ericaceous shrub roots, which I'll show you. But even with that, the bog became a source of carbon to the atmosphere, a source of carbon dioxide and a source of methane. So the response of the plant communities and the increase in nutrient availability was not enough to keep this bog from becoming a source of carbon as the bog warmed up and dried up. And so here's showing you the root response led by my postdoc, Avni Malhotra. She looked at ericaceous shrub root growth over the growing season in 2014 to 2017, looking at it in response to soil moisture. And note this is backwards, so it goes wet to dry, or soil temperature going from colder to warmer, and found that moisture and temperature were both driving this increase in root growth. That is, more roots when it's warmer and more roots when it's drier, which makes sense. So the roots were sort of expanding into this volume of peat that was drying out to take up more nutrients for the plants, but even so, wasn't enough to keep the bog a carbon sink. And I noted that we observed increased nutrient availability, but it was interesting, the response we saw was greater and deeper peat. So let me show you what that looks like. So we used ion exchange resins to look at nitrogen, which is the hotter colors, and phosphorus, which is the cooler colors. And this is in hummocks at 10 centimeter depth and 60 centimeter depth, which is below the rooting zone in this system. We didn't see a response to warming at 10 centimeters in the hummock, but we did see a huge response to warming, an increase in nutrient availability with warming in deeper peat. And so we're currently trying to disentangle. All the sphagnum moss died in our warmest plots, and so does that mean there's sort of a sponge for nutrients? And there, did I do it?
Hit that little button on the...
You told me this was going to happen, and I did it anyway. Here we go. Are they sort of a sponge for the nutrients, or is it like we've seen in other CO2 experiments where there's immobilization by microbes in surface peat in competition from roots, so we don't see the response there? And so we're still sort of investigating what this is, but I think it's cool. All right. So moving even further north to the Arctic tundra. So spruce was a manipulation to create a future that we don't yet see. So to warm up a bog and see what would happen to whether it would become a carbon source. In Alaska and in the Arctic in general, the pace of climate change is so rapid that we don't have to build a future. We're already seeing that it's warming 3 to 4 times faster than the rest of the world, that we're seeing increasing wildfires, that we're seeing increased permafrost thaw and degradation. And so ultimately what we're doing there is tracking the rapid pace of change. I like this picture by Victor Leshik, who is the artist in residence at Northern Arizona University, which I'm pretty jealous. I am an artist in residence. I like this picture because it encapsulates everything that we're doing within the NG Arctic project. So it's federal agency acronyms, but Next Generation Ecosystem Experiments in the Arctic, NG Arctic, previously led by Stan Wolfschlager, who retired last year, and I took over as PI. And we are studying just the everything. So we're thinking about landscape change, thermokarst, slumping. We're thinking about increasing disturbances like wildfire. We're thinking about the march of shrubs across the tundra and how that changes the albedo. We're thinking about permanently frozen soil and how that's thawing, and you have a deepening of the active layer. We're thinking about the melting of ice below ground on the North Slope and the polygonal tundra. And sort of the reason we're thinking about all of those things is because in 2012, when the project started, the Department of Energy asked us to think about doing science in a new way. And that is to do it across institutions. So we have partners at Brookhaven National Lab, Los Alamos National Lab, Berkeley National Lab, and the University of Alaska Fairbanks. And to do things across disciplines. So the observations that empiricists like me make with boots on the ground are going to informing a virtual world of an Earth system model, the Department of Energy's energy exascale Earth system model, the land surface component in particular. And so we fit all of these observations together like puzzle pieces because really our mandate is to understand what is happening across the entirety of the land surface, to make the model represent that appropriately in virtual space, and then to be able to use that model to predict into the future what will happen in the Arctic, because as we all know, what happens in the Arctic doesn't stay in the Arctic. And so as I mentioned, NG emphasizes collaboration among these interdisciplinary teams to incorporate observations into models, and we have lots of partners. And again, funded by the Department of Energy's Office of Science. And I just wanted to mention, I like to tell students that the Office of Science is one of the biggest funders of basic science research in the country, and we don't have like cool NASA shirts, but I feel like we should sometimes because it's sort of important to support a lot of the basic science activities that are happening, including for some folks in the room. So I wanted to sort of show you the places where we're working and a little bit of what that looks like. We started working on the North Slope in 2012, before Barrow renamed themselves Utqiagvik, which means a place to study wild roots, which I appreciate. And then in phase 2 in 2016, we moved further south to include some hill slope tundra on the Seward Peninsula. There's Nome, where the Iditarod ends recently. And there's 3 roads out of Nome, and we work on all 3 of those roads. And really, these 2 different places are to be able to confront the model with data from polygonal tundra, like you saw in the picture, but also hill slopes that have taller shrubs and sort of different hydrology, different glaciation histories, different climate. And you can see, let's see, here's on the North Slope, a warming experiment that our colleague Alistair did. I'm touching permafrost here. With my hands, and then here's further south on the Seward Peninsula. And just to mention that we work across all seasons, and here's Stan being directed to keep digging soil pits. And so I mentioned that, you know, what the Department of Energy is asking that we do is to translate this sort of discovery science from landscapes to disturbance to shrubification into models. And we use models at a bunch of different scales, you know, ultimately headed for the Earth system scale, but using models where we need to represent fine scale processes that the land surface model doesn't yet have or can't do or shouldn't do because they're too fine scale. And then ultimately what we're delivering at the end of phase 3 are 6 different parts of the model, 6 different modules that now look more like the Arctic than they did when we started 10 years ago. And I also wanted to mention one of the things that I appreciate about DOE is that we're mandated to share our publicly funded science publicly. And so they've stood up a data center called ESS Dive, where all data collected through the DOE BER is required to be publicly available. We just got a letter yesterday saying that we have the go-ahead for phase 4 of the project. And so basically in the first 3 phases, we've been converting our measurements from across Arctic Alaska into an improved version of the land surface model. But what we are proposing to do in phase 4 is to take the model on the road. So we know it's better based on observations in Alaska, but we want to see if it can faithfully simulate data from long-term field sites across the Pan-Arctic, like from Siberia, like from Abisko, Sweden, like from Svalbard in Norway, like from sites in Canada. And if it can't, that's great, because we'll know why. And if it can, then we'll be confident to scale up to regional gridded simulations and to Pan-Arctic gridded simulations. And so what we would like to deliver at the end of this phase 4 is confidence in a model that can accurately predict what's happening now, so we can be confident that it's accurately predicting what's happening in the future. And I wanted to show you a little bit about what I do as well. So here's a boardwalk out on the Barrow Environmental Observatory in Utqiagvik, Alaska. This is land owned and stewarded by the UIC Science Native Corporation. And I also like to show folks that, you know, the ice wedges below ground that result in polygonal tundra, you can actually see those polygons on the landscape. And whether or not there's an ice wedge beneath can affect the hydrology, which affects the plant community, which affects the everything. And so it's just really neat to see that patterning on the land surface. If you're a plant ecologist like me, you might think the tundra on the North Slope is a little boring, but you just need to look a little bit closer, because some tundra plant species have 5 times more biomass below ground in an area that is sort of less harsh than the above ground environment there, where the little shrubs are sort of hugging the land surface. And so what we wanted to do was understand the variation in tundra plants, because the model, when we started, represented the entirety of the Arctic tundra with 2 kinds of plants, a deciduous shrub and a C3 grass. And we thought that might not capture the unique characteristics of tundra plant species, and so we wanted to catalog those above and below ground. And so here's 3 of the dominant species on the North Slope: sedge Carex equatalis, which is growing in those troughs right above the ice wedges. Another sedge that grows on drier conditions, and a sister species that makes tussocks further south. And then the shrub that I was telling you about, this is the tallest woody plant on the North Slope. It's this tall. And you can see this makes lots of roots that are pretty deep. This one just has a few roots, but they are gangbusters at taking up nutrients, even from cold soils. And then the shrub species here, if you can see it, associates with an ectomycorrhizal fungi that gives it a competitive advantage in the shallower soil. So we wanted to do a little experiment to think about that. So as George and I were talking about earlier, you know, the prediction for the future is that with warming, you see a deepening of the active layer. And so the question is, will roots take advantage? Will they grow deeper into that active layer to take up nutrients that were previously frozen in permafrost? And that was a question we had too. Where are these plants putting their roots, and where are they getting nutrients from? And so these are the same species I was showing you. This sedge here, most of its roots in gray in the mineral soil, less so in the organic soil, and very few at the permafrost boundary. This sedge here, most of its roots in the mineral soil, same pattern. The shrub that I was showing you, most of its roots in the surface soil. But the pattern of nutrient acquisition is really interesting. So this plant isn't taking up nutrients from where its roots are the most, and this one is. And we think what's happening is that this one's getting a competitive advantage from its ectomycorrhizal fungi to compete with the fungi and the bacteria in the organic soil there. And these sedges are having to put their roots deeper than they might want to. And hardly any of these species have roots at the permafrost boundary, so maybe a thickening of the active layer doesn't really matter as much for nutrient acquisition in the future. We also wrote a paper in 2020, so you can imagine a team of 150 people that spans 5 institutions, that spans disciplines, that has empiricists and modelers, needs to really emphasize safety and a culture of inclusion and sharing and authorship. And so we sort of wrote a paper in 2020 during the pandemic on lessons learned for sort of all of those things, including engaging with the Native communities where we're privileged to do our science. And I'm happy to chat about this too, because I think increasingly we all do team science, and so it's really important to sort of set out those expectations for how folks are going to collaborate with one another before you start and continually as you're doing science. So I've talked to you about 3 places in the world where I work, but those are only 3 places, and I told you that really what we want to do is inform an Earth system model, which is the entirety of the Earth system. And so we put together FRED, which is the Fine Root Ecology Database, in collaboration with Luke McCormick and Schaefer Powell. And I want you to know Schaefer named FRED because he did most of the data input, and he was calling it the fantastic root ecology database, but we didn't think we could publish with that name. And so fine roots are roots that are less than 2 millimeters in diameter, and that seemed to fit just as well. I was telling someone earlier today, we've been trying for years to get models to improve the way that they represent roots below ground and the interface with the soil. And they always told me, you bring me the data, we'll fix the model. And we were like, fine. Put all the data together. And really, I think FRED is useful for a couple of different reasons. It brings together all the data that we need to parameterize models, but it also shows us where the gaps are. So FRED is the largest root trait database in the world. We have 3 different kinds of root traits. I don't want to argue about traits. Traits is a thing you can measure. That's what we decided. And observations from across the globe. But you can see from this map that, of course, because it's a database and because it's an ecological database, there's the coverage is not equal everywhere, right? And so we have a lot more data from temperate regions than we do from arid, polar, or tropical regions, and you can see that here on the map. And we have a lot more data from things that are easier to measure, like root biomass from a core, and less data from things that are harder to measure, like root nutrient acquisition, or root respiration, or anatomy of roots, because you have to cut them open. And so I wanted to mention that really this has sparked some new research. So we have groups like the Tropical Root Trade Initiative, Tropi Roots, and the Arctic Underground, who are working to mobilize data from those different parts of the world. And also I think then the next step will be campaigns that are sort of targeted towards some of the measurements that we're lacking from databases like this. And then for those of you who love fungi, we don't really have anything in the database that is sort of the root fungal interaction. We have the kind of mycorrhizae, but really we need to sort of bring together that sort of partnership, right, into one place so the models can better represent it. So as I've been mentioning, an overarching goal of our work is to inform models that project global responses to changing environmental conditions, and especially feel confident sort of doing that into the future. And I would like to think that this is what it looks like. It's just like zeros and ones floating away from a beautiful tree. But really, this is what it looks like. So these are some of the algorithms that are included in the Earth system model that I work with, right? So thinking about the phenology of root growth, or how many roots are produced per unit leaf, or the depth distribution of roots, or their maintenance respiration. And so just thinking about how I can make observations that can inform some of these algorithms that are the way that these roots are represented in the virtual world. And then another thing that we need to think about when we're thinking about models is how plants are represented in models. So models can't represent the entirety of the 300,000 or 400,000 plant species across the world. They group them into like groups of plant functional types. And so that's like a boreal needle-leaf tree or a deciduous shrub in the tundra. And so really I have to think about how I would group my species into which type and what that means for the way it's parameterized in the model. So I wanted to show you some of the parameterizations. So the current model has 14 plant functional types that are not crops, the 14 that I just showed you. And a couple of the things that the model has to represent what's happening below ground are root carbon to nitrogen ratio and the allocation of roots. And I can just show you this is the root carbon to nitrogen ratio and how it varies across the world. It is 42, so it's the answer to life, the universe, and everything, but it is not variable across any of the plants in the world. And that's true for some of the other parameters too. Allocation of roots to leaves, cellulose fraction, lignin and labile fraction. So you can see, even with the limited data that we have in Fred, we're already making improvements, hopefully, in some of these parameterizations, right? But there's more work to do, of course. So we've got a little work to do, but as long as we keep our eyes on the ultimate goal, which is world domination of roots, yeah. So I wanted to thank, I mentioned that our funding's from DOE, and specifically it's from the Biological and Environmental Research Program, and that includes FACE in the past and NG Arctic Spruce and Fred now. And I also wanted to especially thank the Native corporations where we do our work in Alaska, which is UIC Science, Murray's Igloo, Council, Sitnasuk, and Bering Straits. And then, of course, collaborators and co-authors, of which there are many. And I talked to a couple of folks about science communication today, and I think it's important. You know, people need to know that scientists are people. I think it's cool that you have a museum in the middle of your building. I think I would spend a lot of time there. But we have a really cool communications group in our division, and they've done some podcasts for Fred, for Spruce, and for the Arctic, where you get to hear science, talk about what we do. So thanks, that's all I have. So any questions? Oh, it's a throwable mic. All right.
Takes the department chair to turn it over.
That's why you got hired. Well done. Looks like, oh.
Yeah.
I have concerns.
Oh.
Okay.
I talk into this?
Oh my goodness.
Can I sing? Colleen, this was a wonderful talk, but that lack of variation in C to N ratio and all those other traits, I mean, how do you explain that?
Oh, that was what the modelers decided to do. It is inexplicable, inexplicable.
Okay. Okay, so I misunderstood.
Yeah.
Those aren't real data.
Those aren't real data. So that's where the model is.
Okay, thank you.
Which is easily fixable,
Yeah.
in my opinion.
So basically, the model's a null hypothesis.
Yes. Yeah. But 42 is a good start, I mean, if you're going to start.
Yeah. Thank you.
Yeah. Oh no.
Come on, you can do it.
I want to throw
You can do it. Do it.
3 pointers.
Yeah, nice throw. Yeah, thanks for a great talk. So in one of the, I think it was Spruce, you showed data that everything grew faster later on, but one of the things that was the highest data point was the ambient and growth rate or whatever. I'm not sure what the
I think
Y
that was the root slide. Let me see. You can go backwards and show you. That was the roots.
That's not a tree.
I know what that is.
All right. So this is this the?
No, it
Nope.
was the global warming.
It was like multi-panels, and then there was like not a lot of. Was your the postdoc was working on?
This one. Hang on. This
Yeah,
one.
that.
Yeah.
So the ambient is the circles.
No, those are hummocks and hollows. So we have ambient elevated CO2, but this is the temperature, right? So they're growing more with increasing warming, and there's no CO2 effect yet, is what that's saying. Hummocks and hollows are similar, and CO2 ambient and elevated are similar, but the warming is the big response here. No CO2 effect here, which is weird. We are starting to see it now, though. So it's interesting. There's been a FACE experiment in bog previously, and they didn't see any effect of elevated CO2 on sphagnum growth, for example. And so we weren't sure if we were going to see a CO2 response here, but where we're starting to see it is in the nutrients. So we're starting to see what I think is increased immobilization and elevated CO2 compared to ambient, but that's only in the last 2 years, and these data don't go that far. Does that help?
Yeah.
So
Oh, no.
how do these, now that we are in this graph, how do these graph work? So the variability that you see in soil moisture and temperature, it has to do with the years being warmer and drier?
Yeah, so these are individual years. So this graph is 2014 before the warming started. We only had deep warming here. And then this is when the above ground warming started and continued, and then elevated CO2 started here, and so we hadn't yet seen a CO2. I did not decide to stagger it this way, that's how they did it, but so this is the real temperature of the soil on the bottom. So in the cold plots, it's colder, in the warm plots, it's
That's
warmer.
what I mean. Is
Yeah.
the differences across the years, does it have to do with the duration of the treatment, or it has to do with the particular conditions?
I think it has to do, in my opinion, I think it has to do with when the treatment started. And so we're doing a harvest next year. Next year will be the last year. And so I'm really excited to see in a final harvest where we measure everything and it's been actually warmed above and below ground for 10 years, what the response is. But I think that's one of the interesting thing about experiments like this. So we get to run it for 10 years, right? And you expect the response to change over time. And one of the conversations we had as a team was some people were just saying +0, you know, +2.5, +4.5 on their x-axis. And we as a team decided, no, we need to show the actual temperature and the actual moisture. So that kind of helps a little bit, I think, with what you're saying. It's like responding to that actual variable.
Colleen.
Oh no, you have to
Yeah, I'll talk loud enough.
I'll deliver it. Here, I'm like the MC.
In the work on the Minnesota box,
Yeah.
you had more nutrients available with increasing temperature, but only at 60 centimeters
Right.
depth. So do you think that's because the microbes at that depth don't normally see warmer temperatures, whereas at the surface, you know, it can get pretty warm in the summer.
Yeah.
There. Is that why there's a relationship there and not at the surface?
Maybe. So the reviewers had thoughts about this in the paper, and one of the things they questioned was sort of whether there was degradation of deep peat, which I think is leading to sort of what you're talking about. But another option is water movement down through the peat profile. So maybe there is extra nutrients being mineralized in surface peat, but it's being flushed downwards because water is such a mobile thing in the bog. And so we have the elevated CO2 treatment is depleted in C13, of course, and so we have isotopes of the pore water, and so we can start to look at some of those things. But the other thing we haven't done yet, which Verity is planning to do, is a gross nitrogen mineralization experiment with sort of peat at different depths, because I think it's like increased immobilization by microbes in surface peat, because that's what we've seen before with climate change experiments, but it could be there's more nutrient uptake by more roots, right, which we haven't targeted yet. It could be the sphagnum died and there's like microbes immobilizing nitrogen to decompose the dead sphagnum in those treatments, and so I have questions. I don't have an answer. Yeah.
Thanks.
Yeah. Oh, good. Super.
I have a question on the slide you first pulled up.
Okay. Can do that.
Yeah, this one. So it's interesting that that this is like so dynamic. Like there's not a consistent net primary production over time. Do you have an idea as to like why fine root production, for example, increased and then seems to start to decrease again, or any thoughts on that?
Yeah, it's a great question. I think it goes to your question as well earlier, sort of the dynamic changes. So one of the things that Rich used to like to say was that trees are big and they live a long time, which is true. And so it's nice to do a 12-year-long experiment in the lifespan of a tree because it's longer, right? But what I think happened was in the first year, the carbon was allocated to increase production of wood, which was expected by Rich originally when he hypothesized about it. But production in this stand in particular is limited by nitrogen availability. We did a fertilization experiment next door to show that that was true. And so increasingly then the extra carbon, which is anything above the line, was allocated to the production of the organ that takes up nutrients for the plant, increasingly to roots. But roots are a really nitrogen-rich organ. So you're using the extra nitrogen you take up to make more roots, right? Sort of this vicious cycle. And ultimately, you can see over time, the NPP response to elevated CO2 went away. And we're calling that progressive nitrogen limitation. Ultimately, you don't have enough nitrogen to store extra carbon in the trees that you expected. Now, progressive nitrogen limitation was a really big a hot topic in the literature because everyone was saying it was going to happen and Rich didn't believe it and Duke was saying it was going to happen and it didn't happen at Duke and then it did happen at the FACE site. The other thing we weren't really thinking about back then, but I think would be so interesting, you know, sweet gum are arbuscular mycorrhizal, but the Duke FACE experiment was a loblolly pine plantation, which was ectomycorrhizal, which could mine the soil for nutrients. And so it would be so interesting to think about if it was mycorrhizae driving the different responses to CO2. And that wasn't something that we sort of were thinking to look at then. Does that answer-ish?
Yeah, thank you.
Yeah. Watch out.
In several of your experiments, you have large constructs that potentially influence animal activity. And so I'm wondering whether you've considered the impact of herbivory on some of your results.
Yeah, bunny holes. Yeah. So they had lots of arguments about this as they were building the chambers, maybe even for Occam. Do you remember? On whether they should allow entrances, bunny holes, for bigger herbivores to come in or not. And it was ultimately decided not because they would add variation and uncertainty to processes that were already uncertain. And so we don't study herbivores. We don't have them in the model. My team is trying to convince me to put beavers in the model because they're so important on the tundra, which is true.
Well, even herbivorous insects.
Yeah.
And you know, those blowers might themselves effectively be a fence.
Do you know what the other interesting thing is in the bog? Nobody wants to go in chamber 19 because it is cold +0, but it is elevated CO2, 900 parts per million, and it is a mosquito fest. So nobody wants to go in there and eat mosquitoes. They're definitely going to affect it. The other thing that we've seen is some sort of caterpillar that is feeding on some of the shrubs in the plots, and so for us it's adding variation, but for other folks it's an interesting science question. I just don't think, even with as giant as these experiments are, I don't think we do them at a scale that we can really understand that, or at least not in a way that I would feel confident about. It's not good for my anxiety, Nate.
I'm curious, back to this point, if you guys are thinking at all about soil carbon and whether or not, if these plants are trying to bring in more nitrogen from their roots, if they're exploiting soil reserves and respiring carbon as
That's a great question. That was one of my dissertation chapters. So I was interested to see, so not only are there more roots, they're deeper in the soil profile. And I did a little experiment to show that there was less microbial immobilization compared to mineralization deeper, and so it made sense they were diving deeper. And then, since this CO2 is also depleted in C13, we were able to track the isotopes from the newly produced roots under the FACE experiment into particulate organic matter and mineral-associated organic matter, and we saw an increase in carbon input into the soil at that depth, an increase in carbon concentration at that depth. So it was actually adding carbon to deeper soil.
Cool.
Yeah.
Colleen, as the public facing, I got it just before he said, okay. As the public facing you, what do you think the most important thing is that scientists can tell to the general public about climate change?
Yeah. We had this conversation earlier. What I think the most important thing is, it's real, it's us, but there's things that we can do to mitigate, right? We just have to have collective action to do things in a way that sort of gives us the future we want. And one of the things I was telling someone earlier was that something that Catherine Hayhoe talks about, who's one of my favorite climate communicators, is she says there's so much doom and gloom now that people are getting, they're sort of like tuning out, right? And not willing to do collective action. But have you ever thought about the future that you wanted? And it was funny because I hadn't. I had thought about a bad future. I had thought about the range of bad futures depending on the RCP scenarios. But I never thought about what would I want the world to look like in 30 years and picture that in my mind, or what would I like it to look like in 50 years for my kids. And so that would be my message, is that all of us are motivated people, and so if we think about the future we want, I think we can get there.
Yeah, thank you.
Mm-hmm.
Any final questions? All right, amazing. Go ahead and thank you, Colleen.